Hello and welcome again to this series of molecular biology. Molecular biology. My name is Dr. Adil Abbasi, and today we will be looking at DNA transcription. I hope you liked the last video, which was about eukaryotic and prokaryotic cells. And please continue to like, subscribe, and share. So today we'll be focusing on DNA transcription, right? So one of the reasons is this is a very key mechanism within molecular biology, which is the fundamental basis for life, to be honest, So let's begin. So introduction to DNA. So what does DNA stand for? So DNA stands for deoxyribonucleic acid. And where do we find DNA? We find everywhere, Everyone has their own unique DNA, So DNA helps identify you as a person. You've got your own unique DNA. When there's crimes, for example, DNA is on people's bodies, etc. Fingerprints, etc. Even when we just touching stuff, you've left your DNA there, etc. So that will simplify it down basically. But to look at it from a molecular biology perspective, what is the role of DNA? So DNA stores genetic information which is vital for cell function, development, and inheritance. The structure of DNA. So DNA is referred to as a double helix, which is composed of nucleotides. So the basis of a nucleotide is a sugar phosphate group and a nitrogenous base. Now, within DNA, right, these nitrogenous bases can be adenine, thymine, cytosine, and guanine. So just to fill you in a bit more in detail, so adenine and guanine are examples of purines, which are a type of nitrogenous base, and thymine and cytosine are an example of pyrimidines. So the general rule is for DNA that adenine pairs with thymine and cytosine pairs with guanine, right? And if you look at the diagram here, you'll see the sugar phosphate backbone, and you'll see cytosine pairing with guanine and thymine pairing with adenine. Yeah, and you can see the double helix shape as well. So if you're ever asked about the structure of DNA, it is referred to as a double helix. So what is the functions? The functions are that it stores genetic instructions and facilitates trait transmission during cell division. What do we mean by trait transmission? So by trait transmission, what we mean is genetically inherited diseases, So you share common DNA with your mother and father and your brothers and sisters, yeah? So that DNA can be transferred, and even diseases can be transferred as well, such as sickle cell anemia, haemophilia, alpha thalassemia, beta thalassemia, these are names of diseases that can be transferred during cell division, etc. So moving on. Right, so I want to touch upon this concept which is called central dogma of molecular biology. So the central dogma of molecular biology, it describes the flow of genetic information within a cell. And what that is that's DNA to RNA to protein. So DNA to RNA is a process which occurs transcription, and RNA to protein is a process called translation, but we'll touch upon that in the next video. So DNA encodes the genetic instructions, and during transcription, RNA is synthesized from a specific DNA segment. This RNA then directs the synthesis of proteins through translation. As I said, we'll touch on that in the next video. So the focus of transcription is that this highlights the process of transcription where information in DNA is copied into RNA by RNA polymerase enzymes. So this is very crucial as it initiates the translation of genetic information into functional molecules within the cell. Now, I want to just touch upon this briefly, right, about it being crucial when translation. So if you have a mutation, so some people have mutations in their bodies which can happen as a result of errors during transcription, leading to like genetic diseases or other sorts of diseases. I will touch upon that at the end because I want to go through the flow and make sure we're following A to B, but I will touch upon mutations in this very briefly towards the end. So the overview of transcription is that transcription is a process of copying genetic information of DNA to RNA. This involves 3 steps: initiation, elongation, and termination. Think of initiation as the start of something, and elongation as to making it longer, and to continue it, and termination to stop it. So there are different types of RNA. There's messenger RNA, which carries genetic instructions for protein synthesis. There's transfer RNA, tRNA, which transfers amino acids during protein assembly. There's ribosomal RNA, which builds the structure of ribosomes, which are a site of protein synthesis. And this is all very important to the cell. This helps regulate gene expression, guide cell specialization, and adapt cells to environmental changes. So moving on to what is RNA polymerase. So RNA polymerase is an enzyme which catalyzes the transcription process by synthesizing RNA from a DNA template, specifically messenger RNA. And you have different types within eukaryotes. Remember we discussed last week about eukaryotic cells and prokaryotic cells. We have RNA polymerase 1, 2, and 3, which transcribe different types of RNA. Prokaryotic cells, on the other hand, have a single RNA polymerase. So the function of the RNA polymerase is that it recognizes DNA promoter sequences and facilitates the synthesis of RNA from DNA template. So, as I said before, the steps of transcription. So, the initiation phase. This marks the beginning of transcription. So, it is the start. And during this, you have RNA polymerase binding. So, you have RNA polymerase which attaches to DNA at the promoter region, which is known as the start point for transcription. The DNA strand unwinds and creates a transcription bubble where the RNA synthesis occurs. The key points to understand in initiation is that this is a preparation phase where RNA polymerase binds to the DNA, starting the transcription process, and you get this transcription bubble which allows access to the DNA template for RNA synthesis. Right? One key difference is that within DNA we have adenine pairs of thymine and you have adenine pairs of thymine and thymine pairs of cytosine pairs of guanine. However, in mRNA, when you're doing this transcription process, adenine does not pair with thymine; it pairs with uracil. This is a key difference you must know. It's adenine with uracil. It's only within the DNA to DNA, which is during DNA replication, which I'll make in subsequent videos, that is adenine with thymine. So, the elongation process. So, elongation is known to make it longer. This is the phase of transcription where RNA polymerase produces an RNA molecule by matching RNA nucleotides with the exposed DNA bases. So this results in the creation of a growing RNA strand which is complementary to a DNA template. Let's give you an example. If we have a short DNA strand which is AAGC, during the elongation process we will have UUAG. So say for instance, let's start again. If you have ATGC, you will have UACG, because as I said, adenine pairs of uracil and mRNA, thymine pairs of adenine in both, and guanine pairs of cytosine in both DNA and mRNA. Yeah. So RNA polymerase moves along the DNA template, adding complementary RNA nucleotides, adenine, uracil, cytosine, guanine to form the growing RNA strand. Once again, to reiterate, notice how thymine has been replaced with uracil. DNA unwinds ahead of the polymerase and rewinds behind it. So, termination. During termination, we have recognition of termination signals. So, RNA polymerase identifies various sequences which signal the end of the gene. Now, this is very important, right? So, A triplet of bases is known as a codon, right? And we'll touch upon that in translation. So you will have different stop sequences for mRNA, which will tell you this is the stop, right? So could be the formation of the hairpin structure, could be a UAG, a UAA, etc. Yeah. So the termination mechanisms. The prokaryotic termination involves a hairpin structure forming the RNA, leading to detachment of RNA from the template. The eukaryotic involves different termination, including the cleavage of RNA transcript and release of a DNA template. So, the release of DNA transcript, this involves RNA polymerase detaching from the DNA, releasing a newly formed RNA transcript. So, the key points to remember from this is that termination marks the conclusion of transcription. It ensures the accurate completion of the RNA molecule and detachment of transcription machinery from DNA, allowing the cells to proceed with subsequent processes. Your subsequent processes being translation. So, I touched on briefly about the types of RNA, right? And it's very important that we know about different types of RNA. So you have mRNA, which carries genetic information from the DNA to ribosomes for protein synthesis. You have tRNA, which stands for transfer RNA, and this transfers amino acids to ribosomes during protein assembly, and this is involved during translation. You have ribosomal RNA, which forms the structure of ribosomes and facilitating protein synthesis, and they come in different sizes like 16S RNA and 28S RNA, but we can touch upon that later in subsequent videos. And you have other RNAs such as snRNA, microRNA, and others which play a role in gene regulation, RNA processing, and cellular functions. So snRNA stands for small nuclear RNA. So, something very important to understand as well, yeah. The end of transcription has got something occurring called post-transcriptional modifications. So, what it is once you've had your DNA sequence being transferred to mRNA, so let's think about your DNA transcription sequence as TTT GGG, right? So that is your mRNA that's been formed. So initially it would have been AAA CCC. Now it's UUU CCC, right? So adenine pairs of thymine and guanine pairs of cytosine. Yeah. So, but uracil replaces thymine. Just remember that. Yeah. So that is what you're called pre-mRNA. However, what happens is you've got this immature pre-mRNA molecule, but now it needs to be turned into mature messenger RNA. So you have something called post-transcriptional modifications. So one of these is a 5' cap and a 3' poly A tail. So that is added for mRNA stability and regulation of gene expression. So this is called 5' capping, and the other end is called 3' poly A tail. This is a sequence of A's which indicates the end of transcription. A. And this is used for mRNA stability and regulation of gene expression. Then you have a process called splicing. So, I want to touch about this, about introns and exons. During your mRNA sequence, you have stuff that's called coded DNA and non-coded DNA. Introns are removed during the process of just changing from pre-mRNA to mature mRNA. So, say for instance, you have a triplet of nucleotides, AAA, which is an exon, which is coded DNA, and then you have GGG, right, which is an intron, and then you have CCC, which is again an exon. The intron will be removed, so then you have AAA joining to CCC, yeah, and this will produce a mature mRNA. Introns are always removed, and this is a process called splicing. The purpose of these modifications is to fine tune and make the mRNA molecule from pre-mRNA to a more mature, which enhances stability, allows transport, and helps determine its functional integrity during protein synthesis. Transcription is also regulated as well. So there are many factors which regulate transcription. You have proteins which control the rate of transcription by binding to specific DNA sequences. You have, and these are called transcription factors. So this will turn a gene on or off. Yeah. Then you have enhancers and repressors, and these are DNA sequences that increase or decrease transcription while interacting with specific proteins. Then you have epigenetic modifications such as chemical alterations which influence gene expression without changing the DNA sequence. These are regulating mechanisms are very important because they help continue gene expression, enable cells to adapt to changing conditions, and perform specialized functions. So let's look at differences between transcription in prokaryotes and eukaryotes. So in prokaryotic transcription, you have a single RNA polymerase which performs transcription. This is much more simple, and you do not have introns. mRNA is translated immediately, so there is none of this post-transcriptional modification. This happens only in eukaryotes. So in your eukaryotic transcription, you have 3 mRNA polymerases. You have 3 RNA polymerases. You have RNA polymerase 1, 2, and 3, right? So these have distinct roles. So this is much more complex compared to a prokaryote, and there are multiple transcription factors involved. So the pre-mRNA, this will undergo extensive processing such as your splicing, which is removal of introns, your 5' capping, and your poly-A tail, which is a sequence of nucleotides before translation, a sequence of A's before translation. So this is quite significant, as the understanding these differences is crucial in comprehending the complexities of gene regulation and RNA processing between prokaryotic and eukaryotic cells. So the importance of transcription once again. Regulation of genes can control active genes is crucial for protein synthesis and how a cell functions. Cell specialization. It can direct cells to do specific functions by stimulating distinct genetic programs. Environmental adaptation. This allows cells to respond to changing surroundings by adjusting gene activity. And there's a critical foundation in this. Transcription underpins cellular behavior, developmental pathways, and adaptive capacities, including an organism's resilience and evolutionary success. So, before we go on to the summary of transcription, I just want to touch on something. I said earlier about errors during transcription. So, imagine you have a protein that's corrected. Like, so say for instance you have UCG, yeah. So this will become a triplet in translation, which we'll talk about in the next video. However, if you insert another base or delete a base, what happens is that will change the coding, you know. So if we were to add a UCG, if you were to add an A or delete a G, then the next nucleotide would come in, the next base would come in, and that could potentially alter the whole thing with the mutations. I just want to touch upon that for the next video within translation. This is very important because any sort of errors can lead to mutations, and that will lead to diseases. And this is why some people have certain diseases like sickle cell anemia, or fetal thalassemia, or like even Huntington's disease, etc. But I'll touch upon that in a separate video as well. So the summary of transcription. This involves 3 steps: initiation, elongation, and termination. This converts DNA information into various functional RNA molecules, predominantly mRNA, messenger mRNA. What to take away from this lesson is that transcription has 3 phases. Initiation, which marks the start. Elongation, which helps make the pre-mRNA transcript stronger and produce mRNA. And termination, which concludes the process. Then you have RNA functions. The mRNA carries genetic information. tRNA is involved in protein assembly. Ribosomal RNA helps form ribosomes, and other small RNAs such as small nuclear RNA, microRNA help regulate gene expression. This is quite important for gene regulation, cellular specialization, and adaptability to environmental changes. So, some things to take away is think about the significance of transcription, how this helps shape cellular behaviour, how does it potentially impact development, and how does it drive adaptability. Think about what I said about mutations, and the crucial understanding is that a deep grasp of transcription is fundamental to unravelling the complexities of cellular functions and responses. So, thank you very much for viewing this video. We've come to the end again. The next video will be on translation. I really hope you enjoyed it. Please feel free to leave any comments underneath. Like, share, subscribe. And also, if I made any errors, do feel free to point them out, because I can hear from my nose. I've got a bit of a problem just with my nose, Allergies, is that time of the season. But anyway, I hope you're having a wonderful day, and I hope you enjoyed the video. Thank you very much. Bye-bye.